This is the second lecture, and in this lecture I'll talk about the upstream processing of fermentation and the works or steps that belongs to upstream processing are isolation, preservation and improvement of industrially important microorganism, development of inocula for fermentation, media preparation, instrumentation and process control. So let's start with the first one, that is isolation, preservation and improvement of industrially important microorganisms. And the key works that are done in these steps are isolation of strain, preservation of strain and improvement of strain. First, isolation. So usually we isolate, or not actually usually, I mean where we can find the microorganisms in the nature, okay, in the natural habitat like air, soil and water. So we isolate industrially important or valuable microorganisms from this natural habitat of these microorganisms, okay. And in nature the microorganisms, they live as a mixed populations, okay. So whenever we want to isolate our target microorganisms, we can first we can isolate them, I mean at the first stage we can either isolate them as mixed culture or directly as pure culture, okay. Anyway, whenever we want to isolate our ideal microorganisms, the ideal microorganisms that we choose should contain or possess certain characteristics. These are those characteristics, the nutritional characteristics of the microorganism. Okay, so if the microorganism require a certain type of chemicals which are costly, okay, or which are costly, then it will contribute to the cost of maintaining that microorganism after we isolate that. So it is better that our target organisms nutritional requirement will be not some rare chemicals, okay, not any rare substances. So it will be better if our target microorganism can utilize or live on cheaper available substances and nutritions, okay. Then the optimum temperature of the organism, okay. So at which temperature the organism grow best. So if the required optimum temperature is like too high or too low, it will add up, it will contribute to the final total cost. Because like for example, if the requirement of the microorganism, I mean the optimum temperature requirement is like 40 degrees Celsius, okay, then you have to do all the steps, that means the fermentations at 40 degrees Celsius. So it will be costlier, right? Because you have to keep the condition, I mean, you have to keep the temperature at 40 degrees Celsius, okay. Then the reaction of the microorganism with the equipment to be employed and the suitability of organism to the type of process to be used. Then the stability of organism and its amenability to genetic manipulation. Okay, the microorganism should be stable and it should be easy to genetically manipulate. Okay, so if it is easy to genetically manipulate, then we can use the genetic modification techniques, such as for example the recombinant DNA techniques, to modify it to make it more potential, okay. Then the productivity of the organism measured in its ability to convert substrate into product and to give a high yield of product per unit time. I mean the productivity of organism should be high, okay. And the ease of product recovery from the culture. Now, the isolation method. So, using which method we can isolate our target microorganism. Okay, so or the target organism. So, basically, this refers to the culture method that we use to isolate organisms, and there are different methods such as isolation by stick plate technique, micromanipulator method, enrichment culture method, serial dilution method. Among these methods, the stick plate technique it's actually not good to isolate mixed culture. Okay, so if you want to isolate mixed culture, then you should go for serial dilation, or there are other methods like droplet methods, spread plate methods. Okay, so if you want to use strict plate, in that case, suppose you already know that which species you want to isolate. In that case you can go for strict plate technique. Otherwise, go for serial dilation or droplet method, spread plate method, etc. Okay, and enrichment culture method. Okay, sometimes I mean we enrich the culture. What that means? Suppose I mean the sometimes I mean when the microbes are in the natural habitat. Okay, they can be in a starved condition or a strained condition. That means they can be in weak condition. And another thing is they are in their natural habitat. So when you want to isolate them, you will culture them in the laboratory in a kind of artificial environment, right? So as these microbes have to change their habitat, the conditions are changed. So that's why sometimes they need to adapt with the new condition. Okay, so in that case, I mean, in these scenarios, it is sometimes necessary to enrich them, because if you don't enrich them, then they may get shock during this transfer from the natural habitat to the artificial habitat, and they may die. Okay, so then you will lose your strain. So in that case, sometimes we collect the sample from the natural habitat and put them into media, and enrichment media contains such kind of nutrition or materials which are very easy for the microbes to uptake and enhance their growth. Okay, so then first and okay, so in this method we enrich the culture, and then we go for the next step like strict plate method, or we do serial dilation or droplet methods, etc. Okay, so this was about the isolation methods. So once you have isolated your organism, now you need to preserve it. Okay, so you need to preserve your strain, isolated strain. Okay, so usually I mean, or normally in the laboratories, what many people do, once they have get a pure culture of an isolated strain, they periodically transfer it into a new new fresh media. This is called subculturing. Okay, but the thing is, I mean, you cannot do subculturing for infinite period. Okay, so I mean, if you need a continuous viable microorganisms, in that case you can go for subculturing. Okay, but if you want to preserve for long time, you have to preserve in different way, right? So you can preserve in reduced temperature, for example at 4 degrees Celsius, or a much colder temperature like minus 80 degrees Celsius, or you can store it at liquid nitrogen minus 196 degrees Celsius. Okay, or in the or we can also store them in dehydrated form, like as dried culture or lyophilization. Okay, so in case of refrigerator, I mean, if you want to store at 0 to 4 degrees Celsius, I mean, you will not freeze them. You will keep them at 0 to 4 degrees Celsius. Okay. Usually, agar slant are used. Okay, agar slant are also called agar slopes. Okay. However, please keep it in your mind that you cannot keep the cells for very. I mean, you cannot store the cells at refrigerator temperature for very long time. You can hardly, I mean, for bacteria you can store them for 2 to 3 weeks, or for fungi 3 to 4 months. Actually, why? Because at this temperature, I mean, 4 degrees, around 4 degrees Celsius, the growth of the microorganisms are not completely ceased, are not completely stopped. Okay, rather they grow in a very slow rate. Okay, so as although I mean the growth is very slow, during their growth they will utilize the nutrients, and waste products will be released. Okay, so that means eventually the media condition will be unfavorable for the microbes to I mean for the microbes to survive, so they will die. Okay, So that's why if like if you want to store at around 4 degrees Celsius, please don't keep them more than like for bacteria 2 to 3 weeks. Okay, otherwise the quality of your microorganisms will decline and also they may even die. Okay, so refrigeration is a short. Refrigeration is for short-term preservation, not for long-term. Okay, so if you want to preserve for long time, you should preserve them at very low temperature, like minus 80 degrees Celsius. Okay, or the best is if you can preserve them at minus 190 or minus 196 degrees Celsius. Okay, which is the temperature of liquid nitrogen. Okay, liquid nitrogen is a very famous media to preserve or store organisms. Okay, not only microorganisms, also we store cells like mammalian cells, ES cells or plant cells at this temperature. Okay, so this is actually the best temperature to store the cells. Okay, so and these are called cryopreservation. Okay, if you store your organisms at this temperature, it is called cryopreservation. Okay, and please keep it in mind that like for example, you want to store at 4 degrees Celsius or you want to store at minus 190 degree 96 degrees Celsius. So when you will store your organisms in this different temperature, you cannot use the same media. Okay, you have to store the you have to put your organism or cells into a media, then you will store them. Okay, so the media content or composition for storing in different temperature will be different, right? So if you want to store at 4 degrees Celsius, the regular media composition is good enough. But if you want to store them at minus 196 degrees Celsius, in that case you have to add some extra chemicals there. For example, you have to add some uh, chemicals or agents which will stabilize the pressure. Okay, so usually glycerol are used. Okay, so if we don't use the glycerol, what happened, whenever something frosts, crystals form, ice crystals. Okay, and these ice crystals can damage the cells. If it doesn't matter it's a microbial cells or it's animal cells or plant cells, ice crystal can damage the cells. So that's why we need to add something which will uh, protect the cell from this ice crystals. Okay, so glycerol is a good chemical for this purpose. So if you put glycerol into the media, then they will prevent the formation of ice crystals. Okay, thus the cell will be protected from damage caused by the ice crystals. Okay, so then if you want to, then if you it was cryopreservation. As I said earlier, we can also store the microorganisms in dehydrated forms. Okay, and one way is lyophilization, which is also called freeze dry. Okay, so in case of lyophilization, we have to the culture that or the microbes that we want to preserve are quickly. First, we have to freeze them very quickly at around like very low temperature at around 70 degrees Celsius. Then we will dehydrate them by vacuum. So if you if you put your cells at minus 70 degrees Celsius, this is a very extremely cold temperature, right? So your cells will very rapidly get frozen. Okay, then you have to put them in a vacuum to dehydrate them. So, okay, so ultimately, after this one, the cells they form a powder-like composition. They looks like powder. Okay, and the one good thing about lyophilization is if you lyophilize the cell, you can't store your cells for very long time because it lyophilized form, the cells remain at their dormant state. Okay, and they retain viability for years after years. Okay, for several years. And another good part is you just can keep your lyophilized cell in room temperature. You don't need to keep them at refrigerator. Okay, you can just keep them, put them at room temperature. It also make transportation of the cells very easy. Okay, suppose, okay, so say you stored your cell at liquid nitrogen in that case you have to always keep enough liquid nitrogen into that pot or that space where you have stored the cells. Okay, and also if you want to transfer those cells, you need to use liquid nitrogen. But if you lyophilized your cell, you just can keep them in the room, right, in the open area, just in the room temperature, and you just can easily transfer them. Okay, another way of okay, so another way of storing the cells in dehydrated form is dried culture. Okay, so what is dried culture? Actually, in this method, we can store the soil, the dried soil, the soil that contain the microbes. Okay, so usually the dried soil culture, they are used for preserving mycelial organisms. The organism which form mycelia. Okay, so what we do first, the at first the moist sterile soil are inoculated with the culture. Okay, then we need to incubate them for several days to allow the growth of the culture. Okay, then just uh, allow them to dry at room temperature for 2 weeks. Okay, then we can store this dry soil in refrigerator. Okay, and if you want to keep your microbes or store your microbes at like around 4 degrees Celsius, in the laboratory we frequently do that. In the laboratory we usually store them. If we don't need to store them for a very long time, then we store them at around 4 degrees Celsius. At that time, we usually use agar slab. Okay, so first what we do, we prepare the agar slab, and we inoculate the agar slab with our culture, that is with our microbes, and incubate them for like it can be like you can incubate them for overnight. Like around to incubate for like 16 hours or 20 hours, then just put it in the refrigerator. Okay, however, as I already said, in this method we cannot store the cells for very long term. Okay, so like we can store them for 2 to 3 weeks, so we need to periodically do the subculturing. Okay, that means we need to transfer them into fresh media and then again keep it at 4 degrees Celsius, then again transfer it in new media, then keep it at 4 degrees Celsius. Okay, so this was about the isolation method. In the next part of this lecture, we'll talk about the improvement of stream.